Hi guys, Mrs. Holman here with another science review video. This one is on Punnett Squares. So before I really talk about Punnett Squares, I want to do a quick review of meiosis so that we can understand where these alleles are coming from. So in meiosis, you start with a regular cell with 46 chromosomes. So remember, if this is your parent cell, then it is going to go through interphase, okay, where those chromosomes are duplicated. So remember, this cell has 23 chromosomes from their mom and 23 chromosomes from their dad. Now remember, on each one of these chromosomes, you have a gene. So these would be homologous chromosomes. Here would be your gene. And you can have different alleles for the gene. So in this case, I'm using a big B for brown hair allele and a little b for a blonde hair allele. So during interphase, these are copied. So when these are copied, now we have two big B's and two little b's, okay? So now we have 4 alleles for this trait right here, okay? Then the cells undergo meiosis 1 and meiosis 2, so that now we have one each of these in these 4 cells. So this one might be big B, little b, little b. So each one of these will have one of those alleles for each characteristic. Now this is where we get the combinations that we're looking for in Punnett Squares. Punnett Squares allow us to say, okay, if mom had a big B and a little b to start with, and dad had, say, a big B and a little b to start with, what combinations are possible given this separation, okay? Now, let's remember in fertilization, we have 23 chromosomes in the sperm from dad, 23 chromosomes in the egg for mom. When they come together during fertilization, you get the 46 chromosomes in you, the zygote, okay? So here's the question that Punnett Squares are answering. What is the likelihood that you got this one versus this one? Okay, so let's go through. Now, I'm going to just use the notes that we used in class for this. So if you missed them or if you just want to run through them one more time. So your first step to completing a Punnett square is to determine the genotype of each parent. So in this case, I've given it to you, in addition to this information, what that dominant trait is, what the recessive trait is. But sometimes it may be in the form of a word problem. So for example, give the Punnett cross, give the possibilities given a homozygous dominant female for hair color and a heterozygous male for hair color. So you would have to figure out this information based on that word problem, but here I've already given it to you. Our next step is to draw a box and divide it into 4 squares. Here I've already done that, so there it is. Step 3, write the genotype of one parent along the top. In this case, I'm going to use the male. It doesn't really matter which one you use to go on which side. Okay, so this is the genotype of the male. I'm going to put 1 allele there and 1 allele there. Okay, and then I'm going to copy this going down the page. Okay, 4th step, write the genotype of the other parent along the left side. So again, the other parent is big B, so I'm going to put a big B here and a big B here. And I'm just going to fill those in my following boxes. Fifth step, copy the male's alleles in the boxes going down. So these ones on top, we're copying down. So this big B goes here. That big B also goes here. This little b goes here as well as here. Okay, and then I'm just going to copy that down here. And our very last step is to copy the other parent's alleles in the boxes going across. So here that would be big B there, big B there, big B here, and big B here. And now you are done creating your Punnett square. But we still have to summarize our results. So let's look at our possibilities. Now remember, we already knew what the genotype of the parents were. We are only looking at what is inside the box, okay? In each of those boxes to determine what possibilities we have from this cross. So our possible genotypes, we have either big B, or little b, or big B, little b. Now remember, these two are the same. It doesn't make any difference, but by convention, we write it like this with the big B first. So after we're done making that Punnett square, it's okay to stay like that inside the Punnett square, but I'm gonna write it like this over here, okay? Now notice, there is no little b inside these boxes. So that is not a possible genotype given this cross. Next, we have possible phenotypes. Now remember, phenotype is our physical appearance. All four of these are going to be brown hair. That is because, again, little b is the only one that will show that recessive phenotype, and we don't have that here. So both of these will give us brown hair. What is our possibility of homozygous dominant? So first, let's just go through these. Remember, homo means same. Homozygous dominant, that would be big B, big B. Homozygous recessive, little b. And hetero means different, so big B, little b. So what is our possibility of this genotype given this cross? Well, two of these are big B. So that's going to be 2 out of 4, 1/2, or 50%. So there is a 50% chance of getting a big B genotype given this cross. Homozygous recessive, again, we don't have a little b here, so this is 0 out of 4, or 0%. Heterozygous, big B, little b, we have 2 of those. So that's going to be 2 out of 4, or 1/2, or 50%. Again, a 50% chance of getting a heterozygous genotype. Again, all these three are our genotypes. There's a 50% chance of getting this genotype given a cross between these two parents. And last, here are our two phenotypes. Brown hair, our likelihood is 100%, right? All 4 of these are going to give us brown hair, which means blonde hair is 0% because there is no little b inside these boxes. And then you're done. That is completing a Punnett square. So now we're going to do a couple examples of this to help us through it, okay? So here is where you might want to pause the video in order to try this on your own. So I'm going to be using these SpongeBob genetics genotypes and phenotypes for these examples. So homozygous dominant body shape. So we're going to go to body shape, square pants. So if it's homozygous dominant, homo means same, dominant, that capital letter, that dominant gene, this is going to be big S, big S. Okay? Homozygous recessive, homo is same, recessive is going to be that small one. So this is little s, little s. Okay? So now let's complete our Punnett square. So I'm going to step 1, did that. Step 2, I'm going to draw my box. One parent on top, one on the side. It doesn't matter which one. If you did it the opposite, it just might not match mine exactly, but as long as the ratio of what you have in the box is correct, that's all that matters. So I'm going to put a big S here and a big S there, a little S there, and a little S there. Bring these down, these across. Okay, so now all of these are genotype possibilities. We only have one. All 4 of these boxes are the same. We have 100% big S, little s, which means our phenotype is also 100% square pants. Okay, let's try another one. So, hybrid long nose and stubby nose. Okay, so again, pause the video. Okay, so hybrid. Remember, hybrid is the same as heterozygous. Heterozygous means different. So it's going to be a big L and a little L. Stubby nose, the only way to get a stubby nose is if we have 2 little L's. So notice, I didn't need any qualifiers like hybrid or dominant or recessive. I didn't need any of that. Just by it saying stubby nose, and I know that's the recessive gene, I know it has to be little l, little l. Okay, so step 1 done. I've identified our genotypes of the parents. So I'm going to create my box. That's step 2. I'm going to put one parent on top, one on the side. Okay, again, down and then across. Okay, so for my genotypes, I have 2 that are big L, little l, and I have 2 that are little l, little l. So 50% chance of getting a big L, little l, a 50% chance of getting a little l, little l. Our phenotypes, these 2 will be long nose, 50% chance of getting a long nose, and a 50% chance of getting a stubby nose. Again, notice I said your chances. Punnett squares do not give us the exact results of a cross. It only gives us the likelihood of a particular genotype or phenotype given the genotypes of the parents. Okay, so let's try 2 more. So, purebred round eyes, hybrid round eyes. You can pause the video now. Okay, so purebred round eyes. So remember, purebred is the same as homozygous. So homozygous round eyes is the dominant, so big R, big R. Hybrid round eyes, that's going to be the same as heterozygous, different. We've got a big R and a little r. Okay, so step 1, identify the genotypes of the parents, done. Step 2, I'm going to draw my box. Okay, one parent on top, one on the side. Okay. So here, down, then across. Big R, big R, big R, big R, big R, big R, little r, little r. Okay, again, our genotypes. We have 2 big R, so 50% chance of big R, and 50% chance of big R, little r, based solely on these 4 boxes. Okay, our possible phenotypes. Notice, because there is a big R present in all of these boxes, it will be round eyes 100%. So given these 2 parents, we have a 100% chance of offspring with round eyes. Okay, and the last one, I'm going to show you a little bit of a different way that I may fill it in class. You may see me doing this. It's not really the best way. I really would prefer you guys continuing to go down and across because that's the easiest way to make sure that you get them all right. But if we have any questions about how I do it in class, you will see that occasionally if I'm going fast, I do it in this way. So step 1 is going to be the same. Heterozygous body color, notice these are both the same. So heterozygous for body color would be big Y, little y. Okay, so now we're going to do this cross, and I'm going to show you how I do this. Step 2, again, stays the same. I'm going to make my box. And put my parents on each of these sides. So sometimes I may do it this way. It may help you if you're really struggling with this other way. But again, going straight down and straight across is the best way to make sure you don't make any mistakes. So here in this box, I'm going to take this one and write it there. So this one, big Y, that one, big Y. Here, big Y from there, little y from there. Here we have a big Y and a little y. And here we have a little y and a little y. So sometimes, again, when I go fast, I do it box by box instead of going across and down, or down and across. Either way is fine. Okay, so again, let's talk about the genotypes. Notice, this one was different than any of the other ones we've done so far. This has all 3 genotypes in the box. So we have 25%, one box is big Y, big Y. So 25% chance of getting big Y, big Y. 50% chance, because we have 2 boxes each with big Y, little y. And then a 25% chance of little y, little y. So let's go through our phenotypes. Phenotypes, notice, these each have a big Y, so all 3 of these boxes will be yellow body color. So here we have yellow, 75%. So 3 out of 4 boxes, that's 75%. And blue, 25%. We have a 25% chance of getting a blue sponge given these genotypes for the parents. Okay, so I hope that helped. I hope you guys have a great day. Thank you so much for watching.